Hello, and how are you? Welcome to my session on anatomy and physiology. And today I'm going to focus on deoxyribonucleic acid, what we commonly call DNA. My name is Winnie Barawa, and if you're new here, this is a channel that basically focuses on classwork, lectures, lessons, and also expert interviews relating to healthcare. And if this is an area of your interest, kindly don't forget to subscribe. So stay with me as I take you through this class. And in this class, we're going to explain the structure of the DNA, the base pairing of this DNA, and generally how the DNA operates in our cells. So let's start first. Where does this DNA come from? Or when we mention the DNA, where exactly do we likely going to find it? So in our body, we have the cell, and the cell is basic unit of life. So it is found, in the cytoplasm, and it's found, in our bodies, and it's the basic unit of life, and it has different functions based on where it's located. But at the middle of our cell, we have the nucleus, and it is inside this nucleus that we will find areas like the nucleolus, the nuclear pores, or the nuclear membrane that covers our nucleus. So inside this nucleus, we have the chromosomes. So chromosomes basically has a structure where it has the telomeres and it has the centromeres on its structure. But by simple definition, a chromosome is a condensed state of our DNA. This is a DNA that is under storage, or a DNA that has been stored. So when the DNA condenses into a compact state that will be stored and later can separate and divide to do what we call cell division and replication, in that particular state of storage is the chromosome. So the chromosome, if you unwind it and you open it up, we generate our DNA. Okay. So a segment of our DNA, we call it the gene. So we'll code different genetic characteristics from the DNA. So that's how simple you can identify our DNA. It's found in our cell in a specific location called the nucleus, and it is stored in condensed state that we call the chromosomes. So when the chromosomes, let's describe this DNA. So DNA is responsible for carrying on of hereditary characteristics. Remember, we said a segment of our DNA is called a gene. And the DNA is found in the cell nucleus. It's stored in form of chromatin. So we will see later on that chromatin is part of the chromosome inside it. So it's stored as chromatin, like 30% in the nucleolus. And during cell division, the DNA condenses to form what we call the chromosome. So a segment of this DNA now is what we are calling the gene. So here is a repeat of what we just said again for emphasis. So we have our cell here. Inside the cell, we get to see our nucleus, and in our nucleus, we have the chromosomes. And the chromosome is simply a condensed state of our DNA. And if we uncondense or we open up these chromosomes, we get to see our DNA. And so a segment of the DNA is what we call the gene. Okay, that sounds easy, right? It's not that hard. So going forward, so let's now unwind more about the DNA step by step. So we have said that the DNA is found in the nucleus, and here we have is our nucleus. So this diagram simply focuses on the nucleus, and the basic structure of our nucleus is that in the middle of our nucleus, we normally have an area we call the nucleolus. And then the nucleus is surrounded by a membrane, what we call the nuclear membrane. And the nuclear membrane normally has holes or pathways where materials can exchange in and out of the nucleus, and we call this the nuclear pores. So the nuclear membrane has what we call the nuclear pores. But now inside the nucleus we find nucleolus. So our nucleus here, the one we have, has two sides. There's some light areas and then it has some dark areas. So these light areas, so these light areas, we call them the euchromatins, and the dark areas, we call them heterochromatins. These areas mean something. Dark regions means these are areas that have very dense chromosomes, while the light areas, the euchromatin that is, it means these are areas where it has less coiled chromosomes, meaning there's some active cell division happening. So the euchromatin areas are light areas that indicates to us that there's some cell division happening, and so our chromosomes, are unwinding, or they are opening up to expose the DNA for the division purpose. But the dark areas, that is the heterochromatins, these are areas where the chromosomes are still condensed up, and so they are not dividing, or rather they are still in their storage state. So chromatin, these are chromosomes, strands or strands of DNA that are at different levels of coiling or uncoiling or degree of condensation. It depends on how you want to describe it. So areas which are highly condensed, that is, they are not opened up, we are calling them heterochromatins. But areas that are less coiled, they are not condensed, they have opened up, or the chromosomes are rather opening up and exposing the DNA, we call them the chromatin or euchromatin. So that's how you can describe these two. So let's keep exploring. You've seen there's nucleus, there's nucleolus. So the chromatin, majority of it, you'll find it in the nucleolus, but generally you will find that yes, it has two areas, the dark regions and the light regions. So we've mentioned the chromatin, and we're saying chromatin, this is a condensed state of our DNA. But there's something unique, that when this DNA is condensing, or when the DNA is coiling, it coils around special types of proteins that we call them the histones. Okay. So the DNA does not just coil anyhow, no, it coils around proteins. And these proteins, we call them the histones. So looking at our diagram here, the histones are these areas that look like beads. They look like beads, and when this DNA comes and coils around these beads, although you should not call them beads, you call them the histones. They're histones, so but they look like beads. So when they coil around these histones, they form a structure that we call nucleosome. So a chromosome basically is more of like composition of several nucleosomes. Okay, so many nucleosomes, come together to give you chromatid or chromatin. So the chromatin is a state whereby the DNA has coiled, or it has condensed around histones. But now there'll be many of them. So there'll be many coiled areas of histones. So these areas where the DNA has coiled around the histone, we are calling it nucleosome. So many nucleosomes coming together, they will give you chromatin. Singular, calling it chromatid. So let's look at this. So we have our nucleosome here. We have the first nucleosome, second nucleosome, third nucleosome, fourth nucleosome, like that. So this nucleosome simply means these are areas where the DNA has coiled around histones. But you see, the nucleosome needs to connect to the next nucleosome. So we normally have the linking DNA. So the areas where we are linking one nucleosome to the next nucleosome, we call it the linker DNA. And this now will give us the chromatid. So chromatid simply were these like beads attached on strings, or beads on a string. But this is our DNA, which is now coiling. So every time it coils on a histone, it will be a nucleosome. And as this nucleosome connects to the next nucleosome, we create now the chromatid. So I hope now you're getting this picture. And these chromatids will now condense to give us the chromosome. And then the chromosome will go and stay in our nucleus, in our nucleolus. And the nucleus will belong in our cell, or the central part of the cell. See how it connects. So let's move further and see our nucleosome. How does it look like again? So we are saying the nucleosome is an area where the DNA has condensed around histones, or it has condensed around proteins that we are calling histones. So histones, they're not just proteins per se, but they're unique proteins that can exist as histones of different types. So we have histones type that we call H2As, we have H2B, we have H3, and also we have H4. So we have four main types of histones that are normally used for us to condense our DNA, and they come in pairs. So we normally pair these four types of histones, that is H2A, H2B, H3, and H4. So in short, we will have 8 core histones where our DNA will coil through. Okay, I hope you can see. So we have the brown area here, which are referring to our histones, and there are 8 of them because we are saying they'll be pairing. So we'll have pairing of the 4 main histones. And the DNA will actually coil around it. So the DNA that is coiling on directly on our histones is the central DNA. But remember, we said we need to link the DNA from one nucleosome to the next nucleosome. So a nucleosome simply is definition whereby a DNA has coiled around the histone. So we are linking this one nucleosome to the next nucleosome. So the part of the DNA that is linking us is known as the linker DNA. However, there are some important histones that are very helping us in binding our DNA string so that it doesn't just freely coil. There's a way you need to bind it to stabilize it and to keep it in position. So the histone that is used here is known as the H1. So we have additional other segments that will help us in forming the linkages between the adjacent chromosomes and other histones, and this now includes the histone H1 and the histone H5. Very important to help with binding. So this will come in various pairs. It's not just a single H1 or H5, several of them, and it can go all the way to 48 pairs and so on. But it's just to stabilize and link these histones. So to recap is we are saying we have our DNA that has been coiled to give our main chromosome, and our chromosome simply has a connection of several chromatids or chromatins, and this chromatins is connection of several nucleosomes, and this nucleosomes has coiling of DNA around the histones. Okay, and when these histones have been coiled around, they give us the nucleosome, but the nucleosome will keep connecting to give us the chromatins or the chromatids. Yes, it looks interesting, right? So the organization of our chromatin will be in such a way that it looks like beads attached on a string. So some books you will see this describing it as beads on a string. So let's recall this diagram, and we said the DNA normally gets to coil and condense to give us the chromosome, and so we shall have our chromosome here. But the chromosome is simply a condensation of chromatids or chromatins. And so this condensed state of the DNA is staying in the state of chromatins. If you stretch it out, you will see the nucleosomes. And these nucleosomes are areas where the DNA has coiled around the histones or the histone proteins. And so this is our nucleosome connecting to the next nucleosome using the linker DNA. And as you can see, as it unwinds, you are able to see the histones here. So these histones generally are proteins to help us condense our DNA. And here is our double helix DNA. So the coiling of our DNA in such that it forms some kinds of beads, then we shall describe them as beads on a string. So here is our DNA. And so this DNA will coil around histones. So this is our histone proteins, and then it gives you the chromatin, it gives you the nucleosome. So these nucleosomes will keep condensing and give us the chromatins, and the chromatins will keep condensing and condensing until we end up with a chromosome. Keep moving. So the DNA now has a structure, or rather it has coiled around the histones, and so now we have opened it up and we're looking at the string. So looking at this string, how is the structure of our string? So by description, the DNA is described as a double-stranded helix. In this double-stranded helix has a basic structure, and the basic structure of this chromosome, of this DNA, we normally call it nucleotide, and this nucleotide basically has three main parts. It has a phosphoric acid, where we will call it the phosphate group on the structure. It has a sugar that we call the deoxyribose. So on the structure, we shall call it the pentose sugar. And then it has four main nitrogenous bases, where here on the structure, we are calling it the nitrogenous base. So the four bases are contained includes adenine, where we refer to it as the A, or indicated as A, mostly on the diagrams. And then we have thymine, indicated as T, guanine, indicated as G, and cytosine, indicated as C. So this structure are the basic units of our DNA. So the DNA, in summary, we can say it's made up of a phosphate group that links to the pentose sugar, and it links to the nitrogenous bases. And this structure is what is going to form together to give us our DNA, the famously known double-stranded helix, the double-stranded strands. So the double strand basically is now organized in this way, that the phosphoric acid or the phosphate group and the deoxyribose sugar, they will be forming part of the helical strands, that basically the backbone of our DNA molecule. So the DNA strand is here. These are the strands here. So the DNA strand is a combination of our phosphate group and our pentose sugar, or the deoxyribose. So the sugar and the phosphate will form for us the strand. So looking at these two diagrams, we have the DNA strands, and to just emphasize is that this is the one that contains the phosphoric acid or the phosphate group, and it also contains the ribose or the pentose sugar. So along the strands, we will now attach our bases. So these bases now are the ones that will be pairing. And the pairing of the bases is unique because each base needs to pair to its required base. So the base adenine normally is supposed to pair with the thymine. So A will pair with the T, or rather you can say T will pair with the A. And then the guanine will pair with the cytosine. So G will pair with the C, or the C will have to pair with the G. So this is how the pairing will be happening. So G to C, A to T, and so on. So this is very key because it will help us later on to understand how the genetic characteristics can be generated, how we can do gene coding and genetic generation when we'll be doing replication, DNA transcription, and so on, including protein synthesis. But just to emphasize on the DNA structure, this is how it looks like. We have four nitrogenous bases, that is guanine, cytosine, thymine, and the adenine. And then we have the strand, which is made mostly of the pentose sugar and the phosphate group, which gives us the backbone. So the nitrogenous bases, they lie between these two strands, and they are connected via bonds, what we call the hydrogen bonds. So the hydrogen bonds normally can easily be broken, and it is for this part that the DNA normally can unwind, or it can open up and enable what we call DNA replication. So we'll see how DNA replication occurs later on. Keeping on. So the DNA, because it is double-stranded, it has the sequence of these bases that are pairing in different categories. So the DNA is very important when it comes to genetic coding, especially where we want to code for proteins and amino acids that are very important for protein synthesis. So how do we code for the amino acids, or how do we code for these proteins? So normally we are saying that the DNA has the bases, and bases here we are referring to the four main bases, the guanine, the cytosine, the thymine, and the adenine. So these bases, when you put 3 of them together, we call that a triplet. So where 2 bases have come together, where 3 bases, sorry, have come together, they form what we call a codon. And this codon can also be known as a triplet code. So this code can be used to give out an amino acid or to generate an amino acid. So a gene simply means a composition of triplets of codes that can give us proteins. So a gene, where we called it a segment of a DNA here now, is where we are referring to it as the code. So each code will give you a specific identity for a given amino acid. And amino acids from our protein class, if you've not watched it, you can go ahead, and organic compounds you'll find there in a class we've done on proteins and the different classes of proteins and the building blocks of proteins. We said are simply amino acids. So amino acids are very important because they're the building blocks for our proteins. But where they come from the DNA. And what the DNA does is it organizes our bases in such a way that each 3 bases, they give you a codon or a triplet code. And this triplet code will be used to help us generate different types of amino acids. And so these amino acids will be used to generate different proteins. So the gene consists of all these particular triplets or codes that can be given to code for different proteins or single proteins. So the successive codes or the successive triplets are very important because they will help in controlling the sequence of these amino acids in the protein molecules that will later be synthesized to give us the compounds or the protein compounds. So here's our DNA in this diagram. And in this DNA, we have the bases. So these bases, the 3 bases can actually give you a codon. So like here we have GAG that goes on together to next you find the triple C, then you have TCA and all that. So the DNA cannot synthesize protein on its own. So what it does is the DNA will transcribe what we call transcription. It will transcribe this information into the RNA. So we'll come later to discuss more about RNA and how it takes the information from the DNA. It will transcribe this information into the RNA, where we will have the RNA strand, and the RNA strand will then go to the ribosomes and be translated. So we will translate these codons from the RNA, and from the RNA it will now produce the different amino acids. But the RNA is normally translated in our ribosomes because in the ribosome, that is where the protein synthesis happens, or under cell organelles. If you've not watched that video, kindly go and watch. We have discussed the different organelles and their functions, and the ribosome is where the protein synthesis takes place. So the protein synthesis or translation from the RNA will be happening in the ribosomes, and these proteins will be generated there. So we'll be generating amino acids. All these are different types of amino acids. We have 20 main amino acids in our body, and they're very key. When they're combined together, they give us now the compounds, that is the protein compounds. So keeping on, so we've said that we have 20 main amino acids, and mostly when you're discussing them, we will be using different short forms, but these are our main amino acids. So we have phenylalanine, leucines, isoleucines, methionine, which is known as the start codon. If you want to start the translation or you want to start the coding, you need a start code. This start code is the methionine, the start code. We have valine, serine, prolines, threonine, alanine, tyrosine, and then we have a stop code. Then we have histidine, the glutamine, asparagines, lysines, aspartic acids, glutamic acid, cysteines, arginine, glycines, tryptophans. Now, this can be coded using different types of codes. Okay, And so the genetic codes, mostly I need you to remember that for you to get these proteins, you will code them on the RNA. Okay, so the RNA strands will be transcribed or to be transcribed from the DNA in a process called DNA transcription, which is a temporary procedure, or it's a temporary process that will help you generate the RNA strand. And this RNA strand is what is going to code for us these amino acids. So looking at this, we have triple uracil. Okay, triple uracil. You'll come to see why we have triple U instead of triple A. So on the DNA, normally this is triple A, adenine, and adenine is normally supposed to pair with thymine. But now in the RNA, thymine is normally replaced with uracil. So here you'll have triple U and not triple A, unlike in the DNA. So triple U will give you phenylalanine. You also have W and C, which also can code for phenylalanine. So all this will be different codes for different amino acids, and there's so many of them. But just to remind you is that these are all the basic amino acids that will be used for generation of proteins. And don't worry, we are going to discuss protein synthesis further, or rather refer more to our playlist on anatomy and physiology, and you'll find protein synthesis there. And so that was it, guys, on DNA and its structure. Keep on with us, and if this is a channel you like, the content is really building you up, just remember to like our video, give us a comment, give us an area that you wish you want us to explain more, and share this video with your classmates if you're studying anatomy and physiology, or if you find it so awesome, post it in your socials and let's share. Remember to subscribe and click on the bell so that you get notified when we post our next class. Thank you very much for staying with me all the way to the end. Bye bye, and see you in our next class.